Hi everyone, welcome to the online Bio 181 Exam 2 review. So I do really apologize for making this so late. I was really busy this week, and then last night I had huge technical problems and then everything got deleted and it was really frustrating. But anyway, I'm here now to go through the main topics that you guys wanted me to go through. Basically, this isn't going to be really interactive at all. It's just going to be me talking to you and saying what you need to know for the exam. Once again, I took this class when I was a freshman and I know what Dr. Schaefer expects out of you, so I'm going to probably be giving you more detail than you'll need to know, but my philosophy is always going to be know more and not less, because if you know less, you're going to get it wrong, but if you know more, what's the harm in that? You'll get the question right anyway. So let's begin. So one thing I want you guys to do before we begin here is I want you to take out a piece of paper and I want you to write on the front of it a topic that you really want to go over, and then on the back of it another topic that you really want to go over. And hopefully during this video we're going to get to that topic so you can write notes on this piece of paper and follow along. I always thought that writing on a blank piece of paper was super helpful for me in terms of trying to understand things, so I'm just trying to give you kind of tips on what I did when I was studying for biology exams. So if you want to go ahead and pause the video and do that really quick, you can just start playing the video again after, but I really do recommend taking notes, or at least taking out your own notes and annotating them as we go along in the video, because that's going to help you while you study. And also, please don't forget to sign in. So this video is linked, there's a sign in right above it, hit submit at the end of the video. Please don't forget to do that. I need to log your visits, so please sign in and hit submit at the end of the video. Okay, you guys, so here we have the depiction of the light reactions of photosynthesis. So what I'm going to do is I'm going to go through the light reactions, the dark reactions, and then I'm going to go through glycolysis, then the citric acid cycle, and then the electron transport chain, just so that you can see how we really create energy from the sun. So the first thing I want to point out here is that you don't need to memorize this, okay? The big takeaways that I would get from this is that the photosynthetic electron transport chain, which is what it's pictured here, is making ATP and is making NADPH. Okay, that is the main thing that I would take away from here. They're doing this by a proton gradient, as we all know, and if you can remember what the electron transport chain in mitochondria looks like, you can see that the ATP synthase here is actually flipped backwards. And that's because the proton gradient in this one is actually in reverse. So the high level of protons, so the lower pH, is on the inside, so inside the thylakoid lumen. And so these protons that are in high concentration in the thylakoid lumen are trying to go towards the chloroplast stroma. And they do that via ATP synthase, and that's what facilitates the creation of ATP. So when we get to the mitochondrial electron transport chain, you're going to see that the ATP synthase is actually flipped upside down and that the proton gradient is on the other side. Another key thing to remember here is that you can see that there's two little lightning bolts hitting photosystem 2 and photosystem 1. Those signify photons. So photosystem 2 and photosystem 1 need to be activated by photons in order to work and in order to transport electrons. Another thing to notice here is that we're splitting water. So on photosystem 2, oxygen is being split into water, which is actually something that scientists have been trying to recreate in the lab for a long time and can't, and is really unique for these plants. So overall, we have water splitting, we have a photon, so photoelectric excitation of the photosystems, we're transporting electrons through the chain, hence electron transport chain, and then we're making NADPH and ATP. That's what I would take away from this. And then once again, this is located on the thylakoid membrane. So on the inside you have your thylakoid lumen, and on the outside is the chloroplast stroma. So here we have a depiction of the dark reactions, or more accurately, the light-independent reactions. So when people say dark reactions, that almost kind of implies that, oh, this only happens at night, but that's not really true. So this happens all the time in plants. So even when it's light out, even when it's dark out, it just doesn't require photons, like the photosynthetic electron transport chain that we just went through. The photosystem 2 and photosystem 1 required photon excitation, but as you can see here, there's no photons. We don't need it. We just need carbon dioxide. So you can see the numbers in red on here. We have 6 CO2, 66, 12 C3, which is going to be your P-gals. So, and then 10 C3, 65. So those numbers, the 6 indicates that we are doing this cycle 6 times. Because usually we just start with 1 carbon dioxide and go through the cycle. But in order to create 1 glucose product, remember we need to have 2 excess P-gals. So when we go through this process, we see that at one point we create 12 P-gals, or C3s, just means 3 carbons, which is what P-gals have. And remember that we have to regenerate RUBP every time, and RUBP is a 5-carbon molecule and extremely wasteful. So we have to give, in a 5-to-1 ratio, we have to give 5 of the P-gals that we make back to the Calvin cycle for every 1 P-gal that we can take away. And then we know that P-gals are 3 carbon intermediates, and that if we're going to try and make glucose, we need 6 carbons, right? So we need to make 12 P-gals in order to sneak away 2 of them to get 1 glucose. So the key things that I would remember from this is that you can't just do this cycle once and get 1 glucose. You need to do the cycle so that there's enough times for you to get the right amount of P-gals. Remember that 5-to-1 ratio. So for example, if you made 6 P-gals, 5 of them would go back to RUBP, and then 1 would go back to just floating around in the cytosol, just waiting to be made glucose, right? And so Rubisco is the enzyme that facilitates the Calvin cycle, and it is the most abundant enzyme in the world for a reason, because it sucks. It's horrible at its job. And that's why we have to do the cycle so many times. It's horribly inefficient. And as you can see, we're burning 2 ATP and an NADPH just to do this cycle. So the ATP and the NADPH that we make in the photosynthetic electron transport chain is being used here to essentially make P-gals, and then we take the extra P-gals away and we get to make glucose. So that is kind of the important overall thing to remember about the Calvin cycle. And I would really focus on remembering that 5-to-1 ratio, that for every 6 that we make, 5 of them go back to RUBP and 1 can be taken away. So we need to have the cycle go 6 times to get this to work. And I also wanted to add one thing here. So the Calvin cycle actually happens in the stroma of the chloroplast. So that's where you're going to find the Calvin cycle happening. Also, Dr. Schaefer considers one cycle of the Calvin cycle as starting with those 6 CO2s. So even though it's like you have to regenerate the thing every time, we think about one cycle of the Calvin cycle as being starting with those 6 CO2 because that's what we need to make 1 glucose. So just think of it as one cycle. Okay, so now I want to talk a little bit about photorespiration. As you can see in this diagram, we have the happy side, which is depicting the Calvin cycle of photosynthesis, and then we also have the angry side, which is depicting photorespiration. Now it's angry because it's really wasteful for the plant to accidentally do photorespiration. And so photorespiration is when oxygen reacts with Rubisco instead of CO2. So if you can recall from the last slide, we had those 6 CO2s going into the process in order to make 1 glucose, right? Well, what can happen is because oxygen can also bind to the active site of Rubisco, we can have oxygen bind and actually create this photorespiration cycle in which the plant has to use a lot of energy to regenerate RUBP in order to try and regenerate Rubisco and try to actually do the Calvin cycle like they wanted to in the first time. So basically, it's a huge waste of energy for plants if they accidentally do this. And if their environment is highly oxygenated and there's not enough CO2 around, this can be a real problem for plants. And so, as Dr. Schaefer mentioned in class a little bit, there's actually 2 methods that some plants have evolved to mediate this photorespiration. So one of them is called C4 plants, and he didn't really talk about that one, so I'm not going to go into detail about that one. But the other one is the CAM plants. And so the CAM plants are like our cactuses and the succulents that we have. And so what they do is actually they have these stomata, which are pores on their leaves. And what they can do is that during the day they actually have them closed. And this means that oxygen or carbon dioxide can't get inside the cell or the plant and get to the cells. And so you may think, oh, well, this is bad. They couldn't do photosynthesis. They couldn't do the Calvin cycle because there's no CO2 present. Well, actually, what they do is they open their stomata at night and they capture CO2. So there's there's more CO2 at night because that's when we aren't actively producing oxygen from plants because we can't have the light-dependent reactions running, right? Remember, the water splitting makes oxygen, and so the plant has a higher chance of capturing carbon dioxide at night. Furthermore, opening up the stomata at night when it's not so hot out reduces the amount of evaporation that can happen in these CAM plants. So these plants have just evolved this way in order to try and avoid photorespiration. It actually works pretty well, and they're able to mediate this dangerous and wasteful response. And so the main thing that I would remember from this is that photorespiration is damaging and electrically wasteful and energetically wasteful, and that the body has to go, the plants, not body, the plants have to go and fix it before they can do photosynthesis again. So now in our plant cell, we've gone through the light-dependent reactions, the light-independent reactions, and now we have glucose. Well, what do we do with it? Well, now we do cellular respiration. You may think, oh no, plants do photosynthesis and animals do cellular respiration. Well, in fact, plants have to do cellular respiration so that they could have ATP. I mean, if you think about it, we don't even make ATP in photosynthesis, and so how would the plant cell survive? So plants have mitochondria and chloroplasts, where animals only have the chloroplast, and we don't do photosynthesis, obviously. So you may be looking at this picture and thinking, oh my god, that's really detailed. I don't know any of that. I'm screwed. Don't worry, you won't need to know any of this until you get to biochem, which will probably be your junior year if you're a bio major, or never at all. And so the takeaways that I would take from this is that this is the energy investment phase of cellular respiration. So this is where we actually have to burn some ATP to get ATP out. So if you can see on the little top row, we are burning 2 ATP up there. And that's our energy investment. And if you look at the enzymes, not that you really need to know this, but they're both kinases, which means that we're adding phosphate groups, which helps to activate the glucose and help further along break it down, really. So just know that we are investing 2 ATP here. And then if you see at the top right corner, we have 2 arrows coming out. That's because we get 2 3-carbon products when we break glucose, which makes sense because 6 divided by 2 is 3. So now we have to double everything. From this point on, we have to double everything because we have 2 3-carbon intermediates. So if you look at the next arrow, we see that we are making NADH. But you see that 2 times right next to it, so you have to remember that we're making 2 NADH. And at the bottom, you can see that we are making 2 ATP, but we're making 4 because we have to double it. So let's look at our net outputs, right? So we invested 2 ATP, but we made 4. So our net ATP is actually going to be 2. And then we made 2 NADH, and we didn't use any NADH. So the net outputs from glycolysis are going to be 2 3-carbon intermediates. So it's going to be your phosphates, your 2 phosphates, your pyruvates, your 2 pyruvates, and then it's going to be your 2 NADH and your 2 ATP. So just remember 2, 2, 2, and if you're going to remember ATP, NADH, and pyruvate, you're going to be golden for glycolysis. Alright everybody, so this is the citric acid cycle, or also known as the Krebs cycle. This one is almost like running the Calvin cycle, but in reverse. So remember, in the Calvin cycle we started with CO2 and then we ended with the carbon intermediates that we eventually went on to make glucose. In the citric acid cycle, we're starting with that carbon intermediate and we are making CO2, and we're also making the electron carrier. So we're pretty much doing the exact opposite of the Calvin cycle. The citric acid cycle here has a lot more intermediates than the Calvin cycle, and it's also not horrible at doing its job. It is not as wasteful as the enzyme Rubisco is. So as you can see here, we start with acetyl-CoA. You might be thinking, the end of glycolysis, we had pyruvate and not acetyl-CoA. What the heck is going on? So in between glycolysis and the start of the citric acid cycle, we actually have a thing called pyruvate oxidation, where essentially we are just adding a coenzyme A group and chopping off a carbon. So we put in this 2-carbon, as you can see with the 2 purple circles attached to a coenzyme A, through the citric acid cycle. So Dr. Schaefer usually puts together pyruvate oxidation and the citric acid cycle into one thing. And so if you want to do that, you just have to keep that in mind, what's going on there. But the citric acid cycle itself, strictly speaking, starts with acetyl-CoA. And so just remember that there's that pyruvate oxidation step in the middle that makes acetyl-CoA. And then we are actually also making another NADH in pyruvate oxidation. So pyruvate oxidation is going to get you an acetyl-CoA, and it's also going to get you one more NADH. So just keep that in mind. It is not depicted here, but just keep that in mind because it might affect your NADH count at the end. So with citric acid cycle, we start with the acetyl-CoA, and then we make 3 NADH and 1 FADH2, and at the bottom you see it says GTP there. That is just essentially ATP. So it's like molecularly equivalent to ATP, and so it's technically GTP, but it's ATP. So we are also producing 2 molecules of CO2, which makes perfect sense because we start with acetyl-CoA, which has 2 carbons, and we need to get rid of the carbons but keep the electrons. So if you can remember that acetyl-CoA has 2 carbons, you'll remember how many carbons come out of it, and then I would just memorize that 3 NADH come out of it and that 1 FADH2 and 1 ATP comes out of it. So here I like to tell students something that they've probably never really thought of before. So when you breathe in oxygen, that oxygen is not being converted into carbon dioxide, even though that's what you breathe out. This step right here is what gets you your carbon dioxide. And remember when I said that acetyl-CoA is what gives those 2 carbons needed for the carbon dioxide? Acetyl-CoA comes from glucose. And how do you get your glucose? From the food you eat. So basically, while you're breathing in oxygen to help fuel the electron transport chain, you're breathing out last night's dinner, not just a conversion in your body. And so people like to say here, oh man, if I just start breathing a lot, I'll lose weight. Not really, you'll probably die from hyperventilation first, but it's kind of cool when you think about how the body works and it's not as intuitive as people think. So that's kind of like a fun fact I like to share here. So once again, the main takeaways that I would like you guys to know, and I'm sure Dr. Schaefer wants you guys to know here, is that we make 3 NADH, 1 FADH2, 1 ATP, and then remember, we're gonna be doing this twice because we make 2 acetyl-CoA from 1 glucose, right? We get 2 pyruvate, both of those pyruvates get converted into acetyl-CoA, so you're gonna have to double the products here. So just remember to do that. So what we have going on here is the electron transport chain, which is where we get all of our ATP from, and arguably the most important part of cellular respiration. So just a quick recap, we had glycolysis happen in the cytosol, and then we have pyruvate oxidation, right? That part happens in the mitochondrial matrix because pyruvate gets pumped into mitochondria and then it gets turned into acetyl-CoA, and then citric acid cycle happens in the mitochondrial matrix, and then the electron transport chain happens on the inner mitochondrial membrane, which is the membrane that encompasses the matrix and separates it from the intermembrane space. So the first thing to notice here is that this is extremely simplified. This diagram doesn't even have ATP synthase, which I will show you later, but basically you guys don't really need to know the details once again. Like, you don't really even need to know what cytochrome C does, you really don't even need to know what coenzyme Q does. You probably may not even need to know what complex 2 does. Just know that complexes 1, 3, and 4 are what help to pump the protons, and they accept electrons from our donators, right? So you can see in complex 1 we have NADH becoming oxidized and going back to NAD+, and so the electrons from that are passed into complex 1, and then the proton is pumped into the intermembrane space. So if you can remember from the photoelectric transport chain, the proton gradient was reversed. The low pH is on the inside and the high pH is on the outside, but in this situation, low pH is on the outside and high pH is on the inside, and that's just because ATP synthase itself, it's flipped the other direction in the mitochondria. So we have to pump the protons the other way in order to get a functional ATP synthase. So the black arrows in this diagram show electron movement, obviously, because it's an electron transport chain. If something's moving in it, it's probably electric, which makes sense, electron transport chain, right? So here we get rid of all of our activated carriers. We dump all the electrons into these proteins, they get transferred, and then ultimately these electrons are accepted in their final electron acceptor as water, right? So oxygen is the final electron acceptor, which helps to make water. So if you can remember, the very first step that we do in photosynthesis is split water, and the very last step of cellular respiration, not including ATP making, is making water. So that's kind of like a dichotomy there, if you can remember that. So you could just even think of this as like a complete mirror of the photoelectric transport chain without all the photons, right? So the next thing that we're going to talk about is how the ATP. So now we've reached ATP synthase, right? So as you can see right away, this is flipped from the photoelectric transport chain. You know, the part that makes ATP is on the inside instead of the outside. So we have to make that gradient higher on the outside so that the proton, as shown by H+, can flow through ATP synthase and help drive the formation of ATP. Right here is literally the most important part of cellular respiration. Okay, we make between 32 to 38 ATP here, depending on our body's efficiency status. We almost never make 38. That's just almost a theoretical number, but the middle 30s is about average for ATP making. This is our energy payoff phase. So remember the energy investment phase in glycolysis? This is going to be our energy payoff phase. So the key things really to remember from the electron transport chain is that our complexes 1, 3, and 4 are pumping hydrogens into the intermembrane space, which helps create the proton gradient, which helps to make ATP, the most important part of cellular respiration. So just as a recap, we went from photosynthesis, where we used light energy to make glucose, and now we're using glucose to make ATP. So basically we went from light energy to chemical energy, so glucose, and then we're making a different form of chemical energy in terms of ATP. So this is why photosynthesis and cellular respiration is really awesome, because we can literally take energy from the sun and make ourselves live. It's really cool. So now I want to go over cellular signaling a little bit. So I think it's really important that you guys know the signal type and a basic description of what they do. I'm basically just going to go through this table here and then kind of give you verbal examples of what they do, but I think as long as you know the description and the signal type, that you'll be okay. And then also you need to remember what signal transduction is, which is basically the transfer of a signal that's bound to like an extracellular receptor inside, and signal transduction is like the process that helps cells affect a response in the body. So the first signal type that we have is intracrine. So intracrine is when a cell will produce a signal, so they'll produce a protein that doesn't leave the cell. It'll just go and bind to a receptor that's inside the cell and affect some sort of response. So these are things like transcription factors. So if you remember from the CAP complex, like that CAP protein is sort of like an intracrine signal because it's produced by the body, then it makes a complex and binds to the body's DNA again. So it doesn't leave the cell ever, it stays inside the cell and does things. So autocrine is when a cell will produce a signal and the signal actually leaves the cell, but it doesn't go anywhere. It just goes back and binds right to the cell surface. And so this is kind of like, this is used a lot in immunology. So for example, there's a type of immune cell that'll release a signal called a cytokine. And this cytokine, when they release it, will go back and bind to its own receptors and actually tell the cell to make more of those cytokines and to start proliferating, which just means growing and doing things. So that's like an example of an autocrine signaling. Now juxtacrine is when two cells that have these like receptors that are on the outside touch each other, and this causes signal transduction. So this isn't really like an independent soluble receptor. These are stuck to the membrane, and these cells have to make direct contact in order to have a response. So this is what we see in delta notch. I would definitely know that delta notch is juxtacrine, and I would also probably go back to the diagram and know a little bit about how it works too. So paracrine is when a cell produces a signal that diffuses out of the cell, and then the signal binds to nearby cells. So it's like if somebody sneezed and it was like a really wet sneeze and like people around them kind of felt it after, that's like what paracrine signaling would be. It'd just be like a sneeze on like a group of people in front of you, which is really gross and horrible hygiene, but that's kind of like what paracrine signaling is. And then, you know, the people would obviously react and be like, oh my god, gross man, cover your mouth, whatever, you know, that would be signal transduction. So and then endocrine signaling is going to be when a cell produces a signal that goes into the bloodstream and affects like a lot of cells in your body. So a great example of this is when you go through puberty. So your hypothalamus will send a signal to your anterior pituitary, which sends a signal to the cells that produce testosterone or estrogen, and then the testosterone, estrogen will go into the bloodstream and bind to cells all over your body and tell them to do a number of things that is puberty. So that would be a great example of endocrine signaling. Same with like epinephrine is a great endocrine signaling because, I don't know, like have you ever gotten really scared, like maybe running from somebody who's trying to follow you, or like you almost fall off the bed in your sleep or something and your whole body feels like crap? That's because there's epinephrine everywhere because you got really stressed. So. And then we have pheromonal, which is basically signals that a cell produces that like actually leaves your whole body and binds to receptors on other people's bodies, which I thought this was crazy when I first learned about it and I didn't believe it, but it's totally true. So it's like almost it's like perfume. Like a girl puts on perfume and people around them can smell it, and then people typically like the perfume, right? And so that makes it attracted to them or whatever. So this is like, I don't even know if I can think of an example of this, but I don't think the pheromones are really well understood, so they don't have like concrete examples of them, but it's just like a protein that gets, you know, shot out of your body and binds to receptors in other people's cells. So I guess perfume doesn't really like bind to anything other than your nose receptors, but imagine if like perfume, there were like these perfume receptors on your arms and they did things in your body. That would be like what pheromonal signaling is. So again, the main thing that I would remember from this is that you need to just know the types and the description and maybe like a vague example like the ones I gave.